Hello and welcome. Today we are talking about exchange surfaces. We've already mentioned how substances move into and out of individual cells, but now we're going to look at a problem faced by multicellular living things and in fact larger living things, in that they can't provide enough of the substances they need to all the cells, and they have to have developed exchange surfaces so they can transport things in and out of their bodies and cells. Now, in order for these exchange surfaces to be as good as possible, we need them to have, or possibly have, four features. The first one is to have a large surface area. Second one, to make sure they're thin, so the surface itself is thin. That means there's a short diffusion path and the substance can get in or out as quickly as possible. They need to have a good or efficient blood supply, and that would apply to animals, and sometimes they are ventilated, so this usually refers to air moving in and out or over a surface, and again that's often to do, or usually to do, with animals. Now, the first example we're going to look at for one of these exchange surfaces is the lungs, and here are a set of human lungs, and you may have seen these, or I'm sure you've seen a set of these before in a diagram somewhere. But what we have is some tubules that go into each of a pair of lungs, and these then branch off into smaller and smaller tubules. At the end of every single one of these tubules is a structure that looks a bit like this. So if I was to pull that out of there, enlarge it, you may have seen a diagram that looks a bit like this. This is what we call an alveolus. And the alveolus has the job of carrying out the exchange of carbon dioxide and oxygen with the air. And you can see, if we look at the features of this, the first thing is that we've got our excellent blood supply here. So these all these vessels that are wrapped around the alveolus are all capillaries, and they carry a large supply of blood, very well branched around. I've done part of it in red and part of it in blue, because the blue is deoxygenated blood coming in. It picks up oxygen, becomes oxygenated, and carries it away again to be carried around the body. Now, if we were to look a bit more closely at the structure, if we removed all the blood vessels, just get rid of those, you could see what the structure of the alveolus looks like. And again, if we just kind of sliced it open by removing that part there, this is a diagram that you might be familiar with. It kind of looks like this lobed shape, and the reason why it's shaped like this is to give it a large surface area. Okay, so this shape provides a much larger surface area than if it was just like a balloon shape or a bag shape. Okay, now how you might see this in an exam situation or in a textbook is a bit more just straightforward like this. They might just show you that this area here looks a bit like that, and sometimes they represent it diagrammatically like so. So here are two alveoli, being plural of alveolus, and they're shown slightly differently, but you should be able to explain or describe and explain how this is made, or this makes oxygen and carbon dioxide exchange as efficient as possible. And the best way to do that is to go back to what we were looking at before. Okay, and we said exchange surfaces can be made more effective if they have a large surface area. So yes, these alveoli do have a large surface area. It's to do with the shape of each individual one, and also the fact that there are millions upon millions of those within the lungs. So we have a very large surface area. The actual surface itself is very thin. So if you look here, this blue line here will only be 1 cell thick. This is actually the wall of the capillary in red there, and that will be only 1 cell thick. And so we only have 2 cells to pass through before the oxygen can get into the red blood cells that are found in the blood. Similarly, CO2 moving the opposite direction hasn't got very far to go at all. The third thing is having a good and efficient blood supply, as in animals, and we mentioned that over here when we had the capillaries covering the alveolus there. And the last thing here is that they are ventilated. Now, for the example of lungs, they are ventilated because when you breathe in, we bring in fresh oxygen, and when you breathe out, you actually remove all that CO2 that's been diffused into this space here. Okay, so these are 4 adaptations of the alveoli in the lungs, and you should be able to explain why these are so important to maximize gas exchange. The second example that we need to look at is in the small intestine, and if you were to look at the small intestine, look at it a bit more closely. Here's one part of it. You can imagine that's been cut out, and if you look down it, you see that it's not quite a smooth surface, and if you enlarge it further, you can see that the actual inside surface looks a bit like this. There are lots of finger-like projections that poke inwards where all the digested food is, and these are called villi. One is called a villus, so the singular is villus, but many together are called villi, and they are designed to increase the surface area of the inside of the small intestine so they can absorb as much food as possible, or much digested food as possible. You need to be able to describe it in a little bit more detail than I've just done there. So here I've got a diagram of a single villus. Okay, so that's one of these little red projections that are poking out here. And how do we then talk about the efficiency of this, or how it's made effective as a gas exchange surface? Well, we can go back to our 4 points here. They have a large surface area, yes, because there's lots of them poking out. They're shaped so there's a large surface area. The distance is thin, the diffusion distance. So if you look across here, this is just 1 cell thick. The dissolved food or the digested food has to only pass through 1 cell before it gets to the blood capillary, which can then take it away. A good and efficient blood supply, yes, you can see this dense network of capillaries and are ventilated. This wouldn't be so applicable to the villus because ventilation means just bringing in air, but those are the adaptations that this has. But there is one further thing on the surface of these villi. The actual cells that are covering the villi are actually called microvilli, and these are actually similar finger-like projections, but they're actually on the cells themselves. So we've got all these finger-like projections here, but we've got further smaller ones on the surface of this singular villus there, so that massively increases the surface area. It's probably also worth mentioning here that often in these microvilli we have these little structures here, which you may recognize from B2, and these are mitochondria. And if you're being clever and thinking ahead, why would these mitochondria be here? Well, that's to supply energy, which can be released from respiration, and if they're there, and if we're linking in some topics together, this might be there actually for the process of active transport. And you can imagine that there are substances in the food that have been digested that need to be pulled into the blood by active transport, because you want to get as much of that in as possible, and sometimes that means going against a concentration gradient. Okay, so that's a summary of exchange surfaces. The key thing and the most important thing is to remember these 4 points. Exchange surfaces are more effective when they have a large surface area, are thin to provide a short diffusion path, have a good and efficient blood supply, usually that's to do with animals, well, it is to do with animals, and are ventilated. That's usually something to do with lungs, because we have the movement of air. And if you know those 4, you can often apply it to almost any example that you may be given. But the 2 examples that you do need to know are the ones that we've covered in this video here. Thank you for watching, and I'll see you again soon.